Hello, my name is Paint number 14, and today I'd like to talk about the chapter 6 and chapter 7 of biology. So, chapter 6 first is about the DNA cloning. To work directly with specific genes, scientists prepare gene-sized piece of DNA in identical copies. The process is called as the DNA cloning. So, most of DNA cloning share the same common features such as the use of bacteria and their plasmids. So let's know about the words plasmid and clone genes first. So plasmid are the small circular DNA molecules that replicate separately from the bacterial chromosomes. And the clone genes are used for making copies of the particular genes and producing protein products. So the methods of the DNA cloning: the bacterial restrictions enzyme cut DNA molecules at specific DNA sequence called as the restriction sites, producing a fragment with sticky ends that bond with complementary sticky ends of other fragment. And then the foreign DNA is inserted into the plasmid by using DNA ligase to seal the bonds between the restrictions fragments. Then the combinant plasmid is inserted into the bacterial cell. Then reproductions in the bacterial cells result in cloning of the plasmid, including the foreign DNA called as the cloning vectors. So, this results in the production of multiple copies of the single genes. The genomic library is the collections of recombinant vector clones produced by the cloning DNA fragment from the entire genome. It is made using bacteria. And then a genomic bacteria that is made by using the bacteriophage stores as the collections of the phage clones. And the bacterial artificial chromosomes, or we call it as the BAC, is the large plasmid that can be trimmed down and can carry a large DNA inserts. The BACs are another type of the vectors used in DNA library constructions, and complementary DNA, or the cDNA library, is made by the cloning DNA made in vitro by reverse transcriptions of all the mRNA produced by the particular cells. It represents one part of the genomes, only the subset of the genes described into the mRNA in the original cells. Clones carry the genes of interest can be identified with the nucleic acid probe, having a sequence complementary to the genes. And the process is called as the nucleic acid hybridizations. The probe can be synthesized that is complementary into the genes of interest. And then this is the special part. The use of the culture eukaryotic cells as host cells and yeast artificial chromosome, or we call it as the YAC. So the use of it as a factors help to avoid gene expression problems. So the YAC behave normally in mitosis and can carry more DNA than the plasmid. And the eukaryotic host can provide the post-translational modifications that many proteins require. One method of introducing recombinant DNA into eukaryotic cells is electroporations, applying a brief electrode pulse to create temporary holes in plasma membrane. Alternatively, the scientists can inject DNA into cells using microscopically thin needles. And once inside the cells, the DNA is incorporated into the cell's DNA by natural genetic recombinants. The next method is the polymerase chain reactions, or we call it as the PCR. So the PCR, I will call it as the short name. The PCR, they can produce many copies of the specific target segment of the DNA. The three-step cycle is heating, cooling, and replications. Bring about the chain reactions that produce an exponentially growing populations of identical DNA molecules. So here's the pictures of the technique. So we only have one DNA, not like the whole chain of the DNA. We only have one of them. So like it's a small amount, right? How can we add it and make it into a large amount? So this technique, the PCR, they can help to make it into a large amount, which they can do like copying the DNA. So they separate it into two DNA, and then they make the new strands here. And then they do it again and again and again, so it's gonna add up. So DNA cloning helps DNA cloning helps science and technology fields to develop even more. It allows researchers to, first is compare genes and alleles between individuals. Two, locates genes expressions in the body. And finally is determine the role of the genes in an organism. Another method is the gel electrophoresis and the Southern blotting. So one indirect method of rapidly analyzing and comparing genomes is gel electrophoresis. And the technique is to use the gel as the molecular sieve to separate nucleic acid or proteins by size. The current is applied that causes charged molecules to move through the gel. The molecules are sorted into bands by their size, and then the restrictions fragment analysis the DNA fragments produced by restrictions and some digestions of a DNA molecule are sought by gel electrophoresis. Then the restrictions and fragments analysis is useful for comparing two different DNA molecules, such as two alleles for genes. The protease is also used to prepare pure sample of the individual's fragments. The technique called the Southern blottings combine genes electrophoresis of DNA fragment with nucleic acid hybridizations. So, a specific DNA fragment can be identified by the Southern blotting by using label probes that hybridize to the DNA immobilized on the blots of gel. Another method is the DNA sequence. Relatively short DNA fragment can be sequenced by the dideoxy change terminations method. So modify the nucleotides called deoxyribonucleotide, or we call it short as the DDNTP, attached to synthesized DNA strands of different lengths. Each type of DDNTP is tagged with a distinct fluorescence labels that can identify the nucleotide at the end of each DNA fragment. So you can see that each ends of the nucleotides, they're gonna if like they are the dio dio dideoxyribonucleotides. So if it's this type, the special ones, not the normal ones, they're gonna have the color of their own. Like A gonna be purple, C gonna be pink, and I think C gonna be pink, T gonna be yellow, and G gonna be the green one. So that's just how they can identify which one is like which one by looking at the color of it. So you're gonna know the sequence of it. Okay, so and then we're gonna talk about the expressions of single genes. So change in the expressions of the genes during embryonic development can be tested using first is the Nelson blotting. Nelson blotting combines genes electrophoresis of mRNA followed by hybridizations with the probe of the membrane. And then the and identifications of mRNA at the particular developmental stage suggest proteins function at that stage. Two is the reverse transcriptase polymerase chain reactions, or the RT-PCR, is quicker and more sensitive. Reverse transcriptase is added to the mRNA to make the cDNA, which serves as the template for PCR amplifications of the genes of interest. The products are run on the gel and the mRNA of the interest identified. So both methods are used to compare mRNA from different developmental stages. In situ hybridizations use fluorescence dye attached to the probe to identify the locations of the specific mRNAs in place in the intact organisms. And the automations has allow scientists to measure the expressions of thousands of genes at one time using the DNA microarray assay. So the microarray assays compare the pattern of the gene expressions in the different tissues, the different times, and under the different conditions. And to determine the gene functions. One way to determine their function is to disable the genes and observe the consequence. Using the vitro mutagenesis, the mutations are introduced into the clone genes, altering or destroying its functions. When the mutate genes is returned to the cells, the normal gene's functions might be determined by examining the mutant's phenotype. Genes expressions can also be silenced by using RNA interference, or the RNAi. And since synthetic double-strand RNA molecules match the sequence of the particular genes are used to break down or block the gene's mRNA. Then we're going to talk about cloning plants, which is the single-cell cultures. One experimental approach for testing genomic equivalent is to see whether the differentiated cells can generate the whole organisms. A totipotent cell is one that can generate a complete new organisms. So this is the experiments. Then we're finished talking about the plant. We're gonna talk about the animal. So in animal, we're using things called the nucleus transplantations. So in nuclear transplantations, the nucleus of the unfertilized egg cells or zygotes is replaced with the nucleus of the different cells. Experiments with frog embryos have shown that the transplant's nucleus can often support normal development of the egg. However, the older the donor nucleus, the lower the percentage of the normal developing tadpoles. And about the reproductive cloning of mammals. We're gonna talk about Dolly. So Dolly is a lamb clone from the adult sheep by the nuclear plantations from the differentiated memory cells. It was born in the 1997s, and Dolly premature death in 2003, as well as her arthritis, led led to speculations that her cells were not as healthy as those of the normal sheep, possibly reflecting incomplete reprogramming of the original transplant nucleus. So it is one of the aspirations that Dolly is one of the lamp that makes them know the result of this technique. Then we're going to talk about the medical apply from this chapters. So the medical applications. One benefit of DNA technology is that it can identify in the human genes in which mutations play the role in genetic disease. So those diagnosis of diseases, scientists can diagnose many human genetic disorders by using PCR and primers corresponding to cloned disease genes, and then sequence the amplified product to look for the disease-caused mutations. And the genetic disorders can also be tested by using the genetic markers that are linked to the disease-causing alleles. The single nucleotides polymorphisms, or the SNPs, are used for genetics markers. When the restriction enzyme is added, the SNPs result in DNA fragment with different lengths or restrictions fragment length polymorphisms, or the RFLP. The human genes therapy. Gene therapy is the alterations of an afflicted individual's genes. Gene therapy holds great potential for treating disorders traceable to the single deflection genes. Vectors are used for delivering genes into a specific type of cells, for example, marrow or the pancreas. And gene therapy raised ethical questions such as whether humans' germ-line cells should be treated to correct the defects in future generations. And protein productions by farm animals and plants. Transgenic animals are made by introducing genes from one species into the genome of another animal. Transgenic animals are pharmaceutical factories, producers of large amounts of otherwise rare substances for medical use. Foreign plants are also being developed to make human proteins for medical use also. And next is about the agricultural applications. DNA technology is being used to improve agricultural productivity and food quality. In the animal husbandry, genetic engineering of transgenic animals speed up the selective breeding process, and beneficial genes can be transferred between varieties of species. And in the genetic engineering in plants, agricultural scientists have endowed a number of crops plants with genes for desirable traits. The Ti plasmid is the most commonly used as a vector for introducing new genes into plant cells. And the genes and the genetics engineering in plants has been used to transfer many useful genes, including those for the increasing resistance to pests and others. Okay, next is chapter 7. So we're gonna talk about first is about the Darwin's interest. Darwin's interest in various types of species. Darwin's interest in various types of species was inspired by the stop at the Galapagos Island. In 1844, Darwin wrote an essay on the origin of species and natural selection, but did not introduce his theory publicly. but in June 1858, Darwin received a manuscript from Alfred Wallace, who had developed a theory of natural selection similar to Darwin. So Darwin quickly finished The Origin of the Species, which is the book, and then he published it in the next year. So let's talk about The Origin of Species that Darwin wrote. So Darwin developed two main ideas, which is descent of the modification explains life unities and diversities, and natural selection is the cause of adaptation evolutions. Darwin's note that humans have modified other species by selecting and breeding in individuals with desired traits. The process is called as the artificial selections. So Darwin described four observations of natural, and from these drew two inference. Observation 1. Members of the populations often vary greatly in their traits. 2. Traits are inherited from parents to offspring. 3. All species are capable of producing more offspring than the environment can support. And 4. Overproduction leads to competitions for food and other resources. So the individual best adapts to the environment will be the one who survive and reproduce. Summary of the natural selections. Survival of the fittest. Individual with certain heritable adaptive characteristics will survive and reproduce at a higher rates than the other individual who can't fit and have a certain adaptive. Natural selections increase the adaptations of the organisms to their environment over time, and the speciation. If an environment changes over time, natural selections may result in the adaptations to these new conditions and may give rise to the new species. Natural selections notes that natural selections do not create the new traits, but they edit or select the traits that are already present in the populations. There is evidence that can support the evolutions. First is the anatomical and molecular homologies. Homology is similar results from the common ancestries, and homologous structures are anatomical resemblance, that represent variations on the structural theme, represent instead of the common ancestors. And another evidence is the comparative embryology. Reviews anatomic this one, it's gonna review the anatomical homologies that cannot be seen or is invisible in the adult organisms, which is the vestigial structures are a remnant of features that serve important functions in the organism ancestors. For examples of homologies at the molecular level are genes shared among organisms inherited from the common ancestors. For example, the chicken embryo and the human embryo have the similar functions and the structures. The evolutionary tree. Evolutionary tree are the hypotheses about the relationship among the different groups. It is the Darwinian concept of the evolution tree of life can explain homologies. Evolutionary tree can be made using different types of data, for example, anatomical and DNA sequence data. So here, you can see that from this tree, you can see that they're from one ancestors, they're creating lots of type and species of animals. But it's only the theoretical. Then we're gonna talk about the convergent evolutions. Convergent evolution is the evolutions of similar or the analogous features in the distantly related group. The analogous traits arise when the group independently adapt to the similar environment in the similar way. Next, we're going to talk about the microevolution. It is the change in the allele frequencies in the populations over generations. To process mutations and sexual reproductions, it produces variations in gene pools that contribute to different among the individuals. Populations geneticists measures the polymorphisms in the population by determining the amount of the heterozygotes at the genes and molecular levels. Mutations. Mutations are change in the nucleotide sequence of DNA. Mutations cause new genes and alleles to rise. Only mutations in cells that produce gametes that can be passed to offspring. A point mutation is a change in one base in the genes. The effect of point mutations can vary. First is mutations in non-coding regions of DNA can often be harmless, and mutations in genes might not affect proteins production because of the redundancy in the genetic codes. And mutations in the result in the genes the mutation that result in change in protein productions are often harmful, and mutations that result in change in protein productions can sometimes increase the fitness of the organism and its environment. So meaning that not all the mutations are harmful. And special notes that mutation rates are low in the animals and plants, and mutation rates are often lower in the prokaryotes and higher in viruses. Sexual productions. Sexual productions can shuffle existing alleles into new combinations. In organisms that reproduce sexually, recombinations of alleles is more important than mutations in producing the genetic difference that make adaptations possible. Oh, sorry. I'm so sorry. And gene pools. A gene pool consists of all the alleles for the loci in the populations. A population is a localized group of individuals, or you can call it as a species in the area. That is capable of interbreeding and producing fertile offspring. Next, we're gonna talk about the Hardy-Weinberg equation. The equations for calculating the frequencies of the alleles in the populations. So sorry, let me close the notifications. Okay, so which the P and Q are used to represent the frequencies of two alleles in their populations. The frequency of all alleles in a population will add up to 1. The equation is P plus Q equals to 1. Then we're going to talk about the Hardy-Weinberg principle. The Hardy-Weinberg principle describes an ideal population that is not evolving. The closer the population is to the criteria of the Hardy-Weinberg principle, the more stable the population is likely to be. To calculating the genotype frequencies, the equation is P squared plus 2PQ plus Q squared equals to 1. Hardy-Weinberg ideal conditions. So there is 5 conditions for non-evolving populations are rarely met in the nature. So first is 1 is no mutations, 2 random mating, 3 no natural selection for extremely large populations, and 5 no gene flow. The 3 main factors alter allele frequencies and bring almost every evolution's chain is the natural selection, which is non-randoms, the gene's genetic drift, which is randoms, and the genetic flow, which is also randoms. So genetic drift, it's described about how allele frequencies fluctuated randomly from one generation to the next. The genetic drift tends to reduce genetic variation through loss of alleles. So, in the genetic drift, we have two main reasons that cause the genetic drift. First is the founder effect. The founder effect occurs when the few individuals become isolated from the larger populations. The allele frequencies in the small founders' populations can be different from those in the larger parents' populations. Another one is the bottleneck effect. The bottleneck effect is the sudden reductions in the population size due to the change in the environment, such as like natural disasters, or for example with the wild animals like cheetahs. There's those rich people who like to kill the cheetahs that can cause the bottleneck effect also. If the populations remain small, it may be further affected by the genetic drift. The result, gene pool may no longer be reflective of the original population's gene pools. Another one is we'll talk about three things, right? Natural selections, gene drift, and another one is gene flow. So now we're gonna talk about the gene flow. Gene flow consists of the movement of the alleles among populations. Alleles can be transferred through the movement of the fertile individuals or gametes, for example, pollen. Gene flow tends to reduce difference between the populations over time, and gene flow is more likely than mutations to alter alleles frequency directly. Another one is about the natural selections. So we're going to talk about the relative fitness. Relative fitness is the contributions an individual makes to the gene pool of the next generation relative to the contributions of another individuals. Reproductive success is generally more subtle and depends on many factors. Three modes of the natural selections. One is the directional selections, favor individuals at one end of the phenotypic range. Next is the second one is the disruptive selections, favor individual at both extremes of the phenotypic range. And the last one is the stabilizing selections, favor intimidates variants and act against extreme phenotype. So here are the three modes. Okay, and the sexual selections. Sexual selection is the natural selections for mating success. It can result in sexual dimorphisms, marked difference between the sex, and the secondary sexual characteristic. Males will show due to the mate's choice can increase the male chance of attracting a female while decreasing his chance of survival. Like when the peacock, where the males they're gonna show their fur, like the tail fur to the female for trying to trying to attract the female. Next is about the frequency-dependent selections. In frequency-dependent selections, the fitness of the phenotype declines if it becomes too common in the populations. Selections favors whichever phenotype is less common in the populations. The neutral variations. Neutral variations is genetic variations that appears to confer no selective advantage or disadvantage, for example, variation in non-coding regions of DNA. The origin of species. We have 3 of them. So, the speciations, the origins of new species is at the focal point of Darwin's evolutionary theory. Next is the microevolutions, which consists of the adaptations that evolve within the populations confined to one gene pool. And the last one is the microevolutions, refer to evolutionary change above the species level. Next is about the reproductive isolations. The reproductive isolation is the existence of biological factors, which is barrier, that impede to different species from producing variable fertile offspring. So hybrids. Let's know about the words. Hybrid are the offsprings of cross between the 2 different species. The reproductive isolations can be classified with whether factors act before and or after fertilizations. So all of these are the reproductive barrier between the species. And we can see that we separated into 2 types of barrier. There are prezygotics and there are prozygotic. The prezygotic reproductive barriers. Prezygotic barriers block fertilizations from occurring by impeding different species from attempting to mate, preventing the successful completions of mating, hindering the fertilizations if mating is successful. The prezygotic barriers maintain reproductive isolations and include 5 types of isolations. First is the habitat isolations. 2 species encounter each other rarely or not at all because they are in a different habitat even though they're not isolated by physical barrier. Is the temporal isolations. Species that breed at different time of the day, different seasons or different years cannot mix their gametes. Next is the behavioral isolations. Courtship, rituals, and other behavior unique to the species are effective barriers. Another one is the mechanical isolations. Morphological difference can prevent successful mating. The last one is the gametes isolations. Sperm of one species may not be able to fertilize egg of the another species. The speciations. Speciations can occur in 2 ways. Allopathic speciations, which is the geographic barrier, and a sympathetic speciation, which is the no geographic barrier. So we're gonna talk about the sympathetic first. Oh sorry, I read it wrong. It should be the allopathic. Let me just. Okay, so in the allopathic speciation, gene flow is interrupted, or the microevolution is the cumulative effects of many speciations that extinctions even. When a population is divided into geographically isolated subpopulations, geographic barriers separate the original populations. Separated populations may evolve independently through mutation, natural selection, and the genetic drift. Like these examples. The ancestors, they used to be the same species, but when they get separated by the naturals, they become a different species. Next one is the The next type, as you can see, I have two types. So the next type is the sympatric. In sympatric speciations take place in geographically overlapping populations. The polyploidy is the presence of extra sets of the chromosomes due to the accident during cell divisions. So this is the examples. And we have two types of polyploidy. The autopolyploidy is when an individual with more than two chromosome sets derived from one species. Another one is the alloploidy. It is when the species with multiple set of chromosomes derived from different species. So here. So, and special notes that polyploidy is more common in plants. Many important crops, for example tobacco and wheat, are polyploidy. Next we're going to talk about the last one, the hybrid zone. Hybrid zone is the region in which members of different species mate and produce hybrids. A hybrid zone can occur in a single band where adjacent species meet. A hybrid often have reduced fitness compared with parent species. When closely related species meet in the hybrid zone, there are three possible outcomes. First one is the reinforcement and the strengthening of reproductive barriers, reducing genes flow. Next is the fusions. Weakening of the reproductive barrier with the eventual fusions into one species. And the last one is the stabilizing continued formations of hybrid individuals. This is the examples. So this is the hybrid zone. As you can see, is the zones where the two different species they can meet each others and they mate and producing and a hybrid offspring. Okay, that is all of my notes. Thanks for listening.